Thank you all for attending, and the Biochemical Society and Portland Press are very pleased to welcome you to today's webinar. And this is part of the Biochemistry Focus webinar series, which features research from different areas in the molecular biosciences, as well as practical sessions to support career development. My name is Elton Zeqiraj, and I'm a structural biologist from the University of Leeds. I'm also a member of the Cell Signaling Research Area 5 of the Biochemical Society, and part of our group's job is to suggest exciting and interesting scientific topics for webinars and meetings. So the topic of today's webinar is targeted protein degradation, or TPD for short. And TPD is an innovative chemical approach whereby small molecules are used to induce ubiquitin-dependent degradation of proteins, so that proteins are selectively targeted for degradation by the proteasome. And the TPD is of great interest in drug development, and it can address, amongst other things, inaccessible targets. And it has also allowed us to really reimagine how we think about drug discovery and curiosity-driven research. Our invited speaker today, Dr. Fleur Ferguson, leads a lab at the University of California, San Diego, UCSD, that is working to tackle fundamental challenges in degrader design and discovery using chemoproteomic approaches to understand protein kinase and other enzyme families. Fleur received her PhD in chemistry and chemical biology from the University of Cambridge in 2014 under the supervision of Professors Chris Abel and Alessio Tiuli. And during her PhD, Fleur focused on the generation of small molecule inhibitors of bromodomains. She then joined the Gray Lab at the Dana-Farber Cancer Institute in 2015 to work on small molecule kinase inhibitors and proteolysis targeting chimeras. And in January 2021, Fleur joined UCSD and the SCAG's School of Pharmacy and Pharmaceutical Sciences as an assistant professor. Research in the Ferguson laboratory applies chemical synthesis, biochemistry, mass spectrometry, and cell biology approaches to develop new therapeutic strategies in cancer and neurodegenerative disorders. And the lab's research seek to enable dissection of the cellular signaling networks underlying disease through selective tool compounds that act via targeted degradation and proximity-mediated pharmacology. So today, Fleur will talk about the chemical exploration of targeted protein degradation across the kinome that was undertaken to develop a global map of kinase degradability. And this expansive dataset provides chemical leads for some impressive 200 kinases, with the data made publicly available as an open access chemical proteomics resource. So before I hand over to Fleur, please note that questions will be asked at the end of the webinar after the her presentation, but you can always send them in your questions at any time. You can just type them in the question box, and we will try to answer as many as possible at the end. So I'll now hand over to Fleur, and really looking forward to her talk.
Oh, thank you so much. I am really excited to be taking part in this webinar. Thank you for the invitation and the really kind introduction. As mentioned today, I'm going to be talking about our work mapping the degradable kinome as a resource to accelerate degrader discovery. And so just a quick overview of what I'll cover in this talk. I'm going to introduce our current knowledge, or the knowledge that we had at the time of starting this project, and some common practices in targeted protein degradation. Then I'll describe our experimental approach, how we thought about uncovering some of the rules, guidelines, and investigating some hypotheses in the field. And then I'll discuss briefly how you can actually, you as other researchers can use our degradable kinome dataset to accelerate entry into targeted protein degradation projects. In the second part, we looked at the chemical and cellular variables that may affect targeted protein degradation outcomes. And we also did a brief investigation into the role of the ATPase P97 in targeted protein degradation, and how it affects the molecules and their downstream degradation. And this is really to illustrate how we can use these tools not only to develop selective chemical probes or not only to start new medicinal chemistry projects, but also as tools to interrogate fundamental ubiquitin proteasome biology. And so, of course, can't start a talk about kinases without discussing their long history as attractive therapeutic drug targets. There are more than 35 kinases that are targeted by approved inhibitors, mainly in the oncology space and currently limited to largely receptor tyrosine kinases, but there are a huge number of other kinase inhibitors from other families in phases 1 through 3. And part of the reason why kinase drug discovery has been so successful is that kinases have this shared ATP substrate, which is, of course, small molecule-like, and we can generate leads by synthesizing diverse chemical or small molecules around a privileged scaffold to create a library. And one example is a pyrimidine scaffold shown here. And then screen this library across the kinome either biochemically or using cellular methods such as the chemoproteomic method Kinases to give us these inhibitor profiles. So once you have a library that's well annotated across the kinome, what you can do is pick out a target of interest. Maybe you went to the cancer dependency map and you found a really interesting kinase that's driving your cancer of interest. Maybe a collaborator had some knockdown results in their mouse model. Whatever it is, if you pick a target, you can then have some idea of the initial leads and structure-activity relationships to begin a medchem project where you'll optimize the potency, selectivity, and perhaps the pharmacokinetic properties in vivo. And these probes can be incredibly useful for not only understanding effects on disease and validating targets, but also interrogating the fundamental biology of those kinases which you've optimized molecules for. And many of these compounds, or many of the labs that develop chemical probes, including mine and including the lab where I did my postdoctoral work, will share these probes with the scientific community so that labs of biologists and biochemists who perhaps don't have a lot of synthetic organic chemistry in their group can still use these tools to interrogate their kinase of interest. And so whilst I was a postdoc in the Gray Lab, I was lucky to be able to be involved in lots of these kinase inhibitor discovery projects against both annotated drug targets, developing improved leads with perhaps better potency or better target engagement or selectivity, such as Aurora kinase and PI3K delta gamma. But also a large focus of my work was looking at these understudied kinases which are not functionally annotated and where there are very few publications. And so usually there is some genetic evidence that they may be important in a certain cell or a certain cancer type. And so when we develop these highly selective chemical probes for DCLK1, ERK5, CDK14, and PIP4K, we were super excited to be, really having the first molecules with which to understand these kinases in our hands. But especially for DCLK1 and ERK5, we found when we went and did our downstream investigations in terms of the phenotypes or looking at the cell signaling effects, we were unable to reproduce a lot of the literature or some of the effects that we had seen using genetic studies. And of course, a big reason for this is that a kinase inhibitor does not knock down the entire protein and may not affect other functional domains or scaffolding functions. And so we were incredibly excited to see the development of targeted protein degradation in Craig Cruz's lab at Yale, Alessio Trulli's lab, Jay Bradner's lab, and others as this new technology for essentially engineering the proteome and knocking down the entire protein of interest, really requiring only a small molecule handle with which to pull in the target of interest to an E3 ligase. And so, early work in this field from Jay Bradner's lab targeted this BRD4 protein for which a small molecule was of course described, but by linking this to thalidomide, which is a binder of the substrate adapter cereblon, they were able to show extremely rapid, potent, and highly selective degradation, and of course distinct pharmacology as well. And so this was really an interesting approach and really shows you that you can get different effects from degrading the whole protein BRD4, part of this important transcription factor complex, than you can from just inhibiting a single domain. However, these degraders also come with some initial challenges. So they have a very different type of pharmacology to a small molecule inhibitor. And so our small molecule inhibitors, often we need to optimize for a high residence time, and our drug concentration in the body or in the cells is going to control pharmacodynamics. And as mentioned, we're getting inhibition of enzyme activity. Whereas for degraders have this event-driven pharmacology. So they have catalytic or substoichiometric turnover, and it's the protein resynthesis rate that's controlling the pharmacodynamics rather than the compound exposure. And of course, leading to a complete loss of protein. And so when we think about developing these molecules as medicinal chemists, we have to think about these bivalent degraders a little bit differently in terms of the biochemical assays and the downstream events that we need to be measuring when we're trying to optimize our molecules. Of course, we just mentioned pharmacokinetic and pharmacodynamic relationships. We also have to think about cellular and organismal selectivity because we're co-opting a ligase complex that may or may not be activated, and our cell type of interest may or may not be complementary to the protein we want to degrade. We have to look at the duration of target degradation, as well as a number of other factors, including, for example, understanding how our molecules are working and what their effects are, not just on cellular target engagement, but also on dimerization and ubiquitination. So you really need this whole suite of different assays in order to develop these molecules. And developing this suite of assays, of course, takes time, and understanding which of these are really the key component for achieving degradation is something that we're still figuring out as a field. And so going from this scheme, once you have your assay set up, or once you've decided on your target of interest that you want to degrade, going from this scheme to a targeted protein degrader is not quite as simple as these beautiful diagrams make it look. And perhaps some of you have experienced this in your own medicinal chemistry projects, where you may link all the pieces together and that you have this beautiful modular design, but not every compound is going to work. And this was something that was not only experienced in our own projects, but something that was described by leaders in the field. So we knew that it wasn't just us doing something wrong. And I put some quotes here of this beautiful review, which says, design elements for successful PROTAC-based drugs are currently being elucidated, which sounds very elegant and is kind of translated by Derek Lowe, a scientist at Novartis, in his popular blog In the Pipeline, as targeted protein degradation remains empirical field, which means that you really have to try a lot of things in a lab. And the reason for this is that there are so many variables. And so the different target binder, the different attachment chemistry, the linker length and composition, and the choice of ligase can really affect whether your compound is active. And knowing which one of these variables to go for and which one is the crucial one to change can be a challenge. And as you start to combine these, you get into huge numbers of molecules if you want to comprehensively cover the potential space or the potential chemical space that you maybe should be exploring. And then finally, also you have these cell type specific or target specific variables that may also play into your target's tractability. And so all of this is kind of summed up by this quote from George Breslin and Craig Cruz in their review of where they commented that currently the largest challenge in the use of PROTACs is the length of the discovery phase. And so you have this resource intensive medicinal chemistry component to get to your tool compound with which to do target validation or study downstream effects. And so initial efforts to identify the low-hanging fruit or the tractable targets in the kinome for targeted protein degradation use these highly multi-targeted kinase inhibitors or kinase binders, such as TA684, which is shown here, and in vitro can biochemically bind to over 200 kinases. And a talented graduate student in Nathaniel's lab, Hubert, then treated 2 different cell lines with this degrader that, of course, is based on that multi-targeted binder, was able to observe about 25 of these kinases that were downregulated at a short time point after 1 micromolar treatment. And a similar concept was applied in the Cruz lab as well, in this nice cell chemical biology paper shown here, and where they found a similar finding. And so we hypothesized that these kinases would be highly amenable to targeted protein degradation, and that we could get more selective degraders of them by taking selective small molecule inhibitors that had been developed previously for them and turning them or transforming them into targeted protein degraders. And here's just a subset of the selective kinase probes that were able to be rapidly developed and published in a couple of years following this finding from both the Gray Lab and other labs, and now there are of course many others. And so, encouraged by this, we really were interested in seeing how we could use chemoproteomic approaches such as this one to understand the degradability of the kinome and to really understand the structure-activity relationships across the kinome for targeted protein degradation. Bearing in mind that, after 25 targets, your kinase is maybe likely to not be on that list, given that there are over 700 kinases in the proteome. And so we had all of these outstanding questions, such as how do we prioritize degradable targets beyond this list of 25? How do we rapidly find and optimize leads for targets of interest? What target binder should I use? Which scaffold? Which E3 ligases might be compatible? And there were many, many other questions and hypotheses in the field of targeted protein degradation that we thought we could investigate using a chemoproteomic approach. And so what we did was we created a library of small molecule kinase targeting degraders, really prioritizing different scaffolds and compound diversity here to cover a broad range of chemotypes. We pre-screened these in a cell permeability screening. So this was an E3 ligase target engagement assay, given that we haven't changed the ligase binder. We thought this would be a good proxy for cell permeability, and then selecting for cell permeable molecules with diverse chemotypes. We profiled these using mass spectrometry based global proteomics analysis at the 5-hour time point to give us the best chance of observing direct degradation effects rather than secondary downstream effects that may result from, for example, affecting the cell cycle or affecting transcription. And we chose 1 micromolar as an initial concentration. So we could then screen our library, analyze the data, and get a degradability score, which I'll talk about a little bit later in the talk, but also use this data to generate hypotheses where we could then go in and do more focused medicinal chemistry to systematically explore some of these variables by creating matched pairs and SAR series. So this is the composition of the final compounds that were screened by proteomics, so those that made it through that initial triage. We screened 91 different molecules and we covered a broad range of kinase binder scaffolds. We made sure to incorporate both type 1, type 1.5, type 2 and allosteric binders so that we're able to really understand which conformations of the kinase are amenable to degradation, and if there was any significant difference between these binding modes in terms of compatibility with degradation or with making degraders. We surveyed a range of linkers and we looked at both cereblon and VHL recruiting degraders. And so one thing we didn't do was look beyond cereblon and VHL, and it would be incredibly interesting to kind of go back, especially with some of these more multi-targeted compounds, with all of these exciting new E3 ligase binders that are being described, and see if we can understand the substrate scope or the inducible substrate scope of those ligases. But for this talk, we're really mostly discussing cereblon and VHL mediated targeted protein degradation. So we were super excited to find hits across all clades of the kinome, as you can see mapped out here on this kinome tree. And so we were able to degrade a large percentage of the kinases that we could detect by proteomics in the cell lines we used. And encouragingly, we rediscovered the majority of kinases that had described degraders in the literature at the time, giving us some confidence that the hits we were finding were real. And then we also were able to significantly expand on the number of degradable kinases that we described beyond what was known. And so we also looked at understudied kinases and different types of kinases beyond the protein kinases that you see mapped out on this kinome tree. And we're encouraged to find a number of lipid kinases, metabolic kinases, and other types of kinases where we were also able to degrade, bringing our total up to 220. And so these are kinases that have been nicely described. You can see them here, including pseudokinases, in this beautiful paper from Peter Sorger's lab that's currently on bioRxiv, sort of describing this expanded kinome that is beyond those 500 kinases. And so we were curious as to how often our kinases were being degraded and how that we could use that data to really understand their ligandability or degradability. So what we did was we looked at how many times a kinase was degraded in an experiment in our data set. And what we found was that there were a number of kinases that were frequently degraded and some kinases which were only degraded by 1 or 2 compounds. And so we came up with a degradability score that basically scored how many unique molecules were able to degrade a kinase of interest. We wanted to check that this result was not simply biased towards highly studied kinases, which may have more ligands described in the literature or more potent ligands described in the literature. And as we have developed our library based on compounds from both in-house chemical data, the academic literature and the patent literature, we thought maybe we're just biasing ourselves towards highly studied kinases. So what we did was we took different knowledge metrics that have been developed by the Illuminating the Druggable Kinome Consortium, which is part of the NIH Common Fund initiative. And we took all of these metrics and compared them to our degradability scores or our maximum fold change of degradation that we observed in any experiment. We're pleased to find no correlation, so we believe that this number is reasonably unbiased. But seeing as we saw no correlation there, that sparked us to investigate maybe we have some interesting leads for these understudied kinases, given that about 25% of the kinome is functionally unannotated and has very few publications. So taking this data in reverse, we were able to actually pull out active degraders for at least 16 understudied kinases, including some of those listed here, which could be potentially interesting therapeutic targets. Not only that, but we were also able to find really promising leads that might be a great starting point for a medicinal chemistry campaign. For example, the CDK17 degrader shown here, which only targets or downregulates 2 other kinases under the tested experimental conditions by proteomics. And this kinase binder is of course multi-targeted, and so we would have never have thought to make a CDK17 selective degrader from it, but this empirical data is really showing us that this combination of scaffold, linker, and E3 ligase recruiter actually preferentially targets CDK17 in a way that the small molecule binding doesn't reflect. So we were interested in using this target tractability to see if we could understand it in terms of how can it accelerate our discovery programs. And so what we did was we took a look at these highly degradable kinases, and we pulled out aurora kinase as being one of the most tractable targets for degradation. We then took alisertib, which is a selective aurora A inhibitor, and created these aurora kinase degraders where we're able to rapidly identify potent and selective degraders for this degradable kinase, kind of validating our hypothesis that it was highly degradable. And these validated by Western blotting, where we're able to see extremely potent and almost complete loss of aurora kinase at the 4-hour time point at low 125 nanomolar concentrations. And importantly, these degraders had distinct pharmacology from the parental inhibitor. So this is a viability curve in the MM1S cell line, which is resistant to alisertib, and you can see relatively modest effects on viability, even up to high 10 micromolar concentrations. However, when we treat with our degrader derived from the same ligand, we can see a really different effect and increased potency and maximal loss of viability with that compound. So if you don't just want to do an easy project, or if you don't just want to look at highly studied or highly degradable kinases where you know you have lots of leads, but if you have a particular kinase in mind that maybe is the pathway you study, maybe it has come up in your genetic screen, you can also use this database and mine it to identify perhaps the best scaffold to start your campaign from. And so we decided to see how this would work by taking an example kinase, which is CSK. CSK is involved in STING pathway signaling. So it's a really interesting kinase to look at beyond oncology, also in immunology. However, there are no selective inhibitors described for CSK, and so if you wanted to start a CSK degrader project, it might be really hard to figure out based on what's currently known where to begin from. However, using our database, we were able to find lots of molecules that were able to degrade CSK, and if we examine their proteomic profiles, we identified this DB3291 compound, which was actually a relatively selective CSK degrader. Again, only a few off targets here. And so this, I think, really highlights how empirical data is at this point crucial, but can also be really incredibly informative for beginning medicinal chemistry campaigns, in particular against kinases where you might not have an obvious selective lead, but even those where you may have a number of different scaffold choices to make. This empirical data can help you highlight the one that's most likely to give you a potent and selective result. And so, of course, 220 kinases is far too many medchem projects for one lab or two labs to take on alone. And so we decided to make this data publicly available to try and accelerate degrader projects across academia and industry, as well as, of course, we would love it if people that also did computational and data analysis on this kind of nice uniform data set too. So we wanted to make this easy to use in case you don't have all of the programming and bioinformatics skills that many medicinal chemists are much more focused on, perhaps chemistry or biochemistry. But we want to make this easy to search, easy to use. So this is the website for the tool, and we would, of course, encourage everyone to use it. You can download all the data, you can make your own heat maps and figures, you can move the cutoffs and customize the volcano plots as well. And so, yeah, we're very excited to present this, and we really hope that this will allow people to develop kinase degraders across the kinome for all the targets they're excited about. And I should mention the small molecules are also available on request from Nathaniel's lab too. So if you want to test some of them out, you can request those as well. Okay, so now you have your lead molecule, now you have your initial hit, you have to do medchem optimization. And as we mentioned before, perhaps that is not quite so simple as was described in the diagrams because of all of the different things you could try. And so we were interested in examining some of the hypotheses in the field which are being presented and usually described in the context of one specific project, one scaffold, one cell line, one target of interest, to pull out these guidelines, which it's hard to know how general are these, when do these apply, do these apply across all different proteins or only this one protein of interest, and some of those guidelines were contradictory. And this is incredibly important because the variables that you feel are crucial for compound activity are of course going to influence how you test those molecules and how you try to optimize them. And so we wanted to understand that kinome-wide with statistical rigor. And really kind of figure some of that out. So what we did was we took our kinase degraders that had a large number of targets, so our multi-targeted degraders, and we figured out which combination would be able to target the largest number of unique kinases. So we came up with these 4 molecules that together can degrade over 100 kinases. And so this now allows us in just 4 treatments, 4 experiments to survey a large percentage of the kinome, and we can of course perturb our cells or do different experiments to measure different variables and understand the effects on a broad range of targets to try and see how applicable these hypotheses are. So of course, the first thing we wanted to look at was cellular target engagement. We've talked a lot about how degradation is uncoupled from binding because of this substoichiometric mode of action, but there has to be some binding, and we were really curious to understand how often do degraders significantly engage the target of interest that they're degrading. So what we did was we used our standard global proteomics profiling pipeline to understand the degraded targets in multiple cells. And then in the same cell line where cereblon had been knocked out, we performed a kinative cellular target engagement profiling. This is activity-based proteomic profiling where a covalent ATP mimetic is used to fish out the kinases, and when you treat your cells and they engage that kinase, they compete with that pull-down probe, and the results are analyzed using proteomics. So we did this for our 4 molecules. I'm just showing one example on the left where we plotted the percent inhibition of binding to those pull-down probes versus the relative protein abundance, and you can see that there isn't really a very clear correlation between degrader activity and degrader target engagement. And if we summarize this and look across all 4 of the experiments that we did, we can see that for a large amount of these compounds, we're seeing no significant target engagement, but we're still seeing degradation, indicating that only a low percentage of occupancy is required for active degradation. But there were also plenty of compounds that engaged a target of interest, but didn't degrade it, showing that, inhibition or binding is not enough. You have to, of course, have all of the other steps that go along with protein degradation as well. And then there were a good number of cases where we saw target engagement and degradation together, showing that perhaps this relationship is specific for certain kinases or certain scaffolds. And so by categorizing those, we were able to get a better idea of what that looks like across the kinome. So ternary complexes are a huge part of the discussion around targeted protein degraders because of course they are mechanistically required, but how important is it to have a highly abundant and highly stable ternary complex versus a transient ternary complex that allows for catalysis but perhaps isn't very long-lived or perhaps doesn't have very tight binding. We were really interested to understand what that looked like for our degrader library. And so again, we did global proteomic profiling, this time in HEC293T cells. And we then expressed flag-tagged cereblon and did affinity purification mass spectrometry to understand which compounds were able to induce complexation and enrich a kinase of interest and how that related to which kinases were degraded. So again, here's the results for just one example molecule on the left. We can see in the volcano plot the degraded kinases of this SB1G187 compound. And then we can see in this hockey plot here the enriched kinases that were able to form a complex with cereblon in the presence of this small molecule degrader. And I should mention that these experiments, the affinity purification experiments, were done in the presence of a proteasome inhibitor to prevent degradation, of course, of the complex targets. So what we were surprised to find was that the majority of degraded targets did not have enrichment of the complex in our experiment, indicating that those complexes may be short-lived, may have or may not be very high affinity, which would be consistent with a catalytic mode of action or a transient ternary complex that's highly efficient in mediating compound turnover, but is not perhaps very long-lived or abundant. One limitation of this experiment is, of course, that we are only looking at the 5 hours time point, and we're using an affinity purification where we may be getting rid of some of those weakly bound complexes. So we cannot conclude, of course, that there's no complex because we know that's required for degradation. However, we were able to see, really a minority of the time we could see the complex and we saw degradation, indicating that it may be important to have an abundant complex in some cases, or it may be just a feature of the particular molecules we are using. Finally, we saw a surprising number of cases where we found a ternary complex, but we did not observe degradation in the experiment despite detecting the protein. And so this kind of indicated to us that it's actually possible to form non-productive ternary complexes where perhaps you can bind or you can induce binding with cereblon, but you do not get the subsequent ubiquitination, or you do not get the subsequent degradation that you obviously need for an active molecule. And again, of course, this is looking at rapid degradation at the 5 hour time point. So this we thought was really interesting result. Another thing that can affect complex formation kinetics is the abundance of each of the proteins, so especially your target of interest, but also perhaps your ligase substrate adapter. So we were curious to look at how that would affect the degradation profile. And so what we did was we quantified the relative levels of every protein or every protein detected in the proteome across 3 different cell lines. And then we profiled our multi-targeted small molecule kinase degraders in those cell lines as well. And one of the surprising results that we saw from this was that we saw distinct proteins being degraded in each of the cell lines and different numbers of protein kinases really being hit in these different cell lines as well. And so while many of these were overlapping, which is encouraging, some of them were not. And so you can kind of see that plotted in these Venn diagrams here where we have MALT4 in blue, Kelly cells in orange, and then HEC293T cells are the gray circles. And so this really highlighted to us that especially with degraders based on multi-targeted binders, it's really important to profile the compounds in the context in which they will be studied, and perhaps for in vivo experiments it may be important to look at degradation across different tissues. So having found this, we then went back to ask our original question, which is what is the relationship between target protein expression and degradation? And outside of the extremes, we didn't see a clear relationship between the two, indicating that highly abundant proteins and lowly abundant proteins had a the determining factor in their degradability. We looked across the library, we looked across our whole data set at this as well, taking the average expression in MALT4 and looking at the frequency of degradation, and again, did not see a clear linear pattern. So then we hypothesized that perhaps this could also be related to components of the ubiquitin proteasome system. So we looked at relative levels of cereblon, colon 4A and B, DDB1, as well as the E2 ligases and the P97 unfoldase. And again, we didn't see an obvious relationship between the number of degraded targets and those components of the ubiquitin proteasome system. So another important consideration when we're making a small molecule degrader is which E3 ligase do we pick? And at the time, cereblon and VHL were the most common. And we wanted to understand how this affected the substrate scope and whether a significant number of kinases would be uniquely degraded or preferentially degraded by one ligase over the other. So what we did was we synthesized 3 sets of matched pairs where we kept the linker and the scaffold identical and just switched out the E3 recruiting ligand. What we found encouragingly was, here's a plot on the left where we can see the fold change after treatment with the cereblon degrader versus the fold change after treatment with the VHL degrader. And so what we could encouragingly see is there are quite a number of kinases that lie along that diagonal, which are not preferentially degraded by one ligase over the other. However, there are some clear and obvious examples of ligases which can preferentially degrade one particular kinase. And so if we look across our entire panel of these matched pairs, what we found was that 20 targets could be uniquely targeted by cereblon, and there was 16 that could be uniquely targeted by VHL. And so really by adding an additional ligase of VHL to our kind of degrader toolkit, we were able to increase the targeted space of these 3 chemical scaffolds by 20%. And I think this really provides some interesting experimental quantification of the promise of expanding the number of ligases that we can use for targeted protein degradation, how that may be able to expand the degradable proteome. So we're really excited about this result and to see all of those efforts going on in academia and industry at the moment. So, of course, we cannot forget about the linker. Linkerology has become almost a whole field in itself, and the linker can really affect the compound activity as well as the compound degradation profile. So we synthesized matched pairs which had either an ortho or a meta link to thalidomide and either a PEG 1, 2, or 3 linker, and profiled these multi-targeted kinase degrader panel across the proteome. What we found was that there was, which was really interesting, was that although there were some variances in certain protein degradation, there was this panel or subset of kinases that seemed to be relatively insensitive to linker length or attachment chemistry. And these kinases were those that were enriched for highly degradable kinases in our degradability scoring analysis, indicating that perhaps these kinases may be able to form multiple productive ternary complexes with cereblon, and sort of bringing up the hypothesis that perhaps the plasticity of the ternary complex is something that's a commonality among highly degradable kinases, or the plasticity of the productive ternary complex, I should say. We're also really interested to look at the IMID space or the IMID target space and how this was affected by our linker attachment chemistry and composition, because of course, when we're using IMIDs to recruit cereblon, we may also be recruiting neo-substrates of the cereblon IMID complexes. So what we found was that if we looked at the described IMID targets, we could see some clear structure activity relationships for the IMID based on the linker, with certain kinases preferring, or sorry, certain zinc finger proteins preferring meta attachment, and whereas others clearly preferring an ortho attachment chemistry. We're also curious as to how we could manipulate this in order to get either highly selective degraders that do not target these transcription factors, or multi-targeted degraders with intentionally designed polypharmacology, which may be beneficial, for example, when targeting BTK to also degrade IKZF1 and 3, and there are numerous other examples. So we looked at how our attachment chemistry to thalidomide was affecting the number of IMID targets that we observed across our entire data set, what we found was that if we used an aryl amine or an aminoacetamide, we were highly likely to degrade IMID off targets as well, indicating these are preferred attachment chemistries for intentional polypharmacology and degraders. We're surprised to find that aryl ethers also frequently hit IMID off targets, but that oxyacetamides we discovered rarely hit IMID off targets, and these are really the preferred attachment chemistry for selective degraders, although the presence of 6 degraders which have this oxyacetamide linker but do not, but also degrade IMID off targets indicates that it's still very important to profile these using proteomics to confirm that. So we had done all of this work to annotate the degradable kinome with exciting leads, with hits, with degradability to understand the effects of the important components of that compound in terms of the kinase binder, the ternary complex, the linker length and composition, and the E3 ligase binder, and really categorize the kinome and categorize these targets into the best place to begin using empirical data. But we also realized we had this amazing toolkit for rapidly redirecting almost a fifth of the kinome towards the ubiquitin proteasome system, and we could then study how perturbations on the UPS affected degradation outcomes. And so one of the things we were curious about was the P97 ATPase, which has been described to unfold proteins from large complexes and deliver them to the proteome, but we didn't have any information on whether this was also true for soluble cytosolic proteins such as kinases. So what we did was we treated cells with our multi-targeted degraders in the presence or absence of a P97 inhibitor, and was surprised to find rescue of degradation across the majority of targets. And so you can see here we have the degradation alone in orange, and we have plus P97 inhibitor in blue, and you can see all of those targets moving up. To check this wasn't a general inhibition of degradation, we also did a time course experiment with P97 inhibitor alone and show for these top hits here that there's no effect on the kinase levels in the absence of a degrader. So these are neither upregulated nor downregulated by P97 inhibition alone. We wanted to confirm or examine whether this would be this effect would be impacted by which E3 ligase was transferring that ubiquitin to our target of interest. So we created a series of matched degraders based on GNF7 kinase inhibitor, recruiting cereblon, VHL, or IAP, and could reliably see rescue of degradation in the presence of a P97 inhibitor in all 3 cases, really indicating that the role of P97 in delivering these substrates to the proteome goes well beyond its currently described substrate scope, and also includes a vast array of kinases, such as the 100 or so kinases that we've examined here in this experiment. And so although the role of P97 and the exact mechanism of this, there's still a lot of exciting work to be done, we felt this was an illustrative example of how this toolkit of multi-targeted degraders can actually be used to study fundamental ubiquitin proteasome biology, and we hope that many labs excited about that will request those molecules and use them to study their hypotheses around the UPS system as well. And so just to wrap up, we have created this open access resource of degrader hits and whole proteome profiles for over 220 kinases. We quantified the effects of cellular and chemical variables on targeted protein degradation outcomes across the kinome. And we used multi-targeted degraders to uncover a dependency on the P97 ATPase for kinase degradation. This project was a massive collaborative effort and a whole bunch of fun to work on. I have to specifically acknowledge Catherine Donovan in the Fisher Lab, who was a co-lead on this project and did a large amount of the proteomic profiling, along with chemists in the Gray Lab, biochemists in the Fisher Lab who helped along the way with all of the biochemical testing, donated molecules from their projects were perhaps inactive against target of interest, or some additional compounds were donated by Tabo's lab, Sarah Burlage's lab, Lee Tan's lab, and of course Irene Gabriel's lab helped us with that MM1S blood cancer experiment. So we had a bunch of fun working on this. It was a massive team effort. We of course need to thank our funders as well. Finally, thank you all for your attention. I'm happy to take any questions.
It's great. Thanks a lot, Fleur, for that great presentation. It's really, really nice to see such a massive endeavor And I'm glad to see that there's a really good resource out there for lots of chemical tools that can be used. And I hope that all of our attendees get a chance to go and do some data mining at the website. So at this point, so we've had a few questions and during your presentation, and please keep those coming. And I wondered if I could start with perhaps the first question would be about the linker length and linker length and composition. And somebody's asking whether does linker length matter more with one ligase versus the other, or in other words, which ligase is more tolerating to linker length and composition, if you have any ideas?
Yeah, that's a really interesting question. And so in our experiment, we actually only looked at cereblon recruiting degraders. So we don't actually have any data on that. But what we didn't do was mine out, the linker length versus the plus. We didn't do linker length plus ligase and look at the number of targets across the database, where although you don't have identical numbers and you don't have identical match pairs, you may be able to get some of that information. So that could be something really cool to look at in the data set that we haven't really thought about yet. And of course, it would also be really interesting to do that experiment again with VHL. Not measured yet, but maybe we can get some initial indications from looking at that data. And we did make some nice tables with all of the different components of in the supporting information of our paper. We actually tabulated, all of the chemical properties, not just like log P and TPSA and so on, but also the ligase, the linker length, and the parental scaffold. And so that might be a quick one to actually go and look at after this talk. Maybe I'll go and do that.
Mm-hmm. Great. Since we're in the topic of the ligases, so there's obviously multiple reports of new ligases, and there's a lot of excitement about making use of those for targeted protein degradation. And I wondered, with how much do you think the additional ligases will contribute to selectivity or potency of degradation, or is that, if you had the choice, which one would you be looking forward to improving? Or
yeah, I mean, this is a really good question. I think we were super excited to see just how significantly adding VHL to that subset of 3 compounds, just simply making 3 more compounds could expand the target space of those scaffolds. And I think that that's an important consideration, is that an active degrader, all of those components are contributing to the activity against those targets. And so I think that we thought 20%, that's pretty compelling, because if we can get another 20% from a different ligase, awesome, but we don't have the guarantees or the information to know yet how those different ligase binders are going to affect that, how many targets they're going to be compatible with. So that's something we need to experimentally determine, but I'm really, really excited about that. I think something that we learned from this project is that it's actually quite difficult to make a highly multi-targeted degrader that directly degrades a large number of kinases. And so although there were many multi-targeted kinase scaffolds, we didn't find all of those as multi-targeted degraders. Some of those were inactive, some of those only degraded 1 target or 2 targets, as we showed in some of those nice examples in the beginning. And so what we're hopeful of is that we've described these 4 or 5 scaffolds where we can robustly see multi-targeted kinase degradation across different ligases, and we hope that that will also be really useful information for people wanting to chemoproteomically survey new E3 ligase ligands, where they can now say, okay, well, I know that this kinase binder has been pretty good for cereblon and VHL in terms of creating a multi-targeted degrader to survey the landscape of what's compatible with my ligase. Can we use this to also accelerate understanding the compatibility of new ligase binders? And so I think that's something we hope that it's a useful piece of information, or we hope can be useful in understanding that question and getting the experimental data, which I think is what we need. And we'd all love to know what the most exciting new ligase is, and just how broad is target space. But I think anything we can do to get there faster and to get there more efficiently is, that was our goal.
Right. So we have one question from Abigail about asking about. Hi there, I was wondering if Dr. Ferguson has any insights, views on targeting phosphatases as opposed to kinases and the relative success one might achieve by comparison.
Yeah, I mean, this is an interesting question, which target classes are amenable to degradation, and does this approach really, work for other targets beyond the kinome, which have had so much success in drug discovery and have so many ligands. I think we're sort of starting to see from the literature that, for example, bromodomains are also highly tractable, and perhaps some other target classes as well. And so understanding that in the context of phosphatases or any other target class of interest is something that, obviously we didn't address here, but if there are, methods for developing multi-targeted degraders of those, or methods for using chemical genetic methodologies such as the degradation tag system, to kind of get into the tractability of either a target of interest or, or across a number of targets, I think that's something that you've got to do experimentally. And I haven't seen any phosphatase degrader papers out there, but I don't think that means it cannot work. I think maybe it just means that it hasn't been published yet, or hasn't been worked on yet.
Yeah, that's very exciting to see if we can reproduce this into other families. And I wondered if we have another question from Julian. Great talk, thank you. Are there any potential mechanisms of resistance that could develop, perhaps by upregulation of DUBs?
So there's been some really beautiful work on resistance mechanisms to degraders done by both Georg Winter's lab and also Nathaniel Gray's lab, looking at degrader resistance mechanisms in cells, which are treated, of course, long-term with a degrader of interest, and then looking at the effects of that cell as they become resistant, which have shown, both both changes in the UPS, but also changes in perhaps the target binding site as well in the context of, CDK12 degraders, for example. So I believe in cellular context, people have examined what are the potential resistance mechanisms. Of course, the clinical resistance mechanisms we still have to find out from the ongoing clinical trials and the use of these drugs in the clinic. But I think, of course, like any small molecule modality, there are potential resistance mechanisms that could occur. And then, it's how do we get around those, or how do we prolong the life of our degraders? We're going to have to be kind of smart about that, but some of that data we're going to have to get from the clinical results, I guess.
Great, thanks. I think so. I'm going to ask probably one last question, if you don't mind, and that perhaps to do with the kinase complexes, and whether in your data sets you actually saw any evidence that when you have a degrader, your kinase gets targeted. But how about kinases that are in obligate partners with obligate partners? Do those partners get degraded as well, or is it really that specific? And I guess this is a general question for other, not just for kinases in particular, but is that, I wonder what
so that's an interesting question. And sometimes we do. So we see, the binding partners, for example, of Aurora kinase. We do see downregulated. We do see some cyclins downregulated when we degrade CDKs as well. But we don't always see the binding partners degraded. And so sometimes they are, and sometimes they are not. And so whether that is because we're looking at a short time point, and perhaps that's a secondary destabilization, whether that's because they're actually stable in the absence of their binding partner, or whether there is another specific or protein specific method for each or mechanism for each different protein, that's something still needs to be determined and looked into further. But sometimes we see co-degradation or co-destabilization, and that is actually, the whole proteomic data sets are are there. And so if there is a particular binding partner you're interested in, you might be able to, well, you will be able to just search it and see if one of the compounds downregulates it. But we don't have a complete understanding of when and why we occasionally get co-degradation, but not always.
Yeah. Great. Well, thank you very much, Fleur, and that's been a really interesting webinar. And thank you for sharing your work today, and for especially at such an early hour in San Diego. And thank you to our audience for listening and asking interesting questions. You can, of course, continue the conversation online. You can follow the Biochemical Society and Portland Press Publishing, and the Twitter handles are on your screen, and you can use them in your discussion about today's webinar. If you have an idea or for a topic to be included in our webinar series, you can submit a proposal on our website at this address, and you can submit your idea to view. You can view your previous webinar recordings and also see what's coming up in these series. So next time in the Biochemistry Focus webinar series on the 7th of July, we will welcome Professor Gurjal Besra, who is the winner of the 2020 Morton Lecture, to present his medal on the role of lipids in the synthesis of the Mycobacterium tuberculosis cell envelope. And you can register for free now on our website. Lastly, I want to highlight that it's more important than ever to stay connected. Joining the Biochemistry Society's community of researchers and specialists is a great way to keep in touch with your fellow molecular bioscientists. Members can take advantage of a variety of benefits, including discounted registration fees for society courses and meetings, exclusive access to a wide range of grants and bursaries, and also personal online access to 2 journals. And so you can visit the Biochemistry Society, biochemistry.org website to find out more. And one final thank you to everyone, and goodbye from us. Thanks a lot.